everyone, welcome to Girls STEMpede. Our guest today is Sydney Floryanzia, a chemical engineering Ph.D. student and a research scientist. Hi, Ms. Floryanzia. Thank you for being here today.
Hi, nice to be here. You can totally call me Sydney.
So before we get into our actual questions, I wanted to ask you an icebreaker, which is if you could meet any female in STEM from history, who would it be and why?
It'd probably be Mae Jemison. She's a fellow Black female engineer, chemical engineer actually, who also went to space. And so I'd love to talk to her about kind of how her chemical engineering background helped her there. I've always known chemical engineering could be applied anywhere. So that's really cool. And also she was really into dance as well as engineering and STEM. And I also have a very strong connection with music and STEM. So I'd love to talk to her about kind of how she sees those things working together as well. And she's not too far back in history. She's still out today making moves and stuff. So maybe one day I will meet her. And she's also a doctor, so she just does it all, like, honestly.
I feel like the cool thing about STEM or science is that, like, in a lot of cases, once you do one thing, once you pursue one thing, everything is so interconnected that it's maybe not like extremely easy, but there's like a pathway for you to travel to go to another career, which is why so many people in STEM are, doctors, and then also chemical engineers are also working with space because they're all very interconnected. My first question for you is, could you briefly tell us what you do, and could you take us through your typical day? So what's your favorite part, your least favorite part, etc.?
Yeah, so I'm a first year PhD student. And so my schedule is pretty variable now. It'll change a lot as I progress in my PhD. But right now as a first year, I have class heavy weeks where I have like testing things. I need to focus on my classes. And I have like lab heavy weeks where I'm in the lab most days. So my class heavy weeks, I go to class, I might have a test, I have seminar, they typically bring in like distinguished scientists in the field to come talk to us and we learn. So it's like a day full of science. And it's really actually cool because at the graduate level, you're really learning about the same thing, but from a lot of different angles. And so looking at the same thing from a lot of different angles, you get a really clearer picture, which is really cool to see. I prefer my research heavy weeks. Monday might be extracting brains, slicing them, extracting brains from rats, slicing them, putting them in little petri dishes. The next day I might come in and the equivalent of if the brain slice was a toddler, changing its diaper and giving it a new bottle. So basically like changing the food that it's in, removing the waste, babying, and they're little babies, they're little squishy babies. So then we'll maybe run an experiment on them. Our most common one is oxygen glucose deprivation. So basically we cut off the oxygen and food supply, which provides like an injury similar to stroke in the tissue model. And then later on, we fix them in formaldehyde and stain them. So that way we can put them under a fancy confocal microscope, shoot lasers at it, and different cells will glow different colors. And we can actually see, did cells die or did they change in response to the stroke injury that we caused a couple days ago? Did their shapes change, all this kind of stuff? And then you write it all down in your notebook as you're going along. So that's my research right now. It'll definitely shift and change over the next few years, but that's me now.
That's so cool. The oxygen deprivation thing sounds really interesting. And you repeat the experiment a lot with different samples of brain, or how does that?
Yeah. So, I mean, I'm still in like my very rudimentary stages, but there's been noted differences in the male and female brain, the brain at different ages. So you have like one control, you make these different changes, how long you subject it to the oxygen glucose deprivation, how much time you give it for recovery, how long after the initial extraction you expose it to the deprivation. So there's a lot of different things you can change even within this very rudimentary experiment, like not rudimentary, but within this very like simple seeming steps experiment, there's a lot of areas for variation. Much to test.
That's awesome. Thank you.